together with the human body, as opposed to exogenous microorganisms that could be ingested, for example, through the diet, or that we may get exposed through the environment, and so on. Okay? So first, we will have like a quick overview of what the discipline of microbial ecology is, what it means to study these ecosystems. And then we'll discuss the two larger ecosystems that are associated with the human body, and actually they both reside in the gastrointestinal tract, and they are the oral microbiota, so the community of microorganisms that live within our mouth, and the gut microbiota that is localized all along the intestinal tract. There are very, very low number in the more proximal regions like the esophagus and the stomach, even though there are still some detectable microorganisms there. And then the density increases as we go along the intestinal tract. The more distally we go, the more microorganisms there are per gram of intestinal content. Okay, so these two microbial communities are the largest communities that are associated with the human body, but they are not the only ones, as we will see in a few slides. So what is microbial ecology? So this discipline studies the relationship between microorganisms and the environment where they are located. So it studies how microorganisms come together and they are able to live together, as opposed to what may happen like in a pure culture in the laboratory, right? So what happens to microorganisms when they have different species of microorganisms where they have to share the same environment, and also it studies the relationship of these microorganisms as a community with the environment where they reside. So all the factors of the environment that may affect the physiological situation in which these microorganisms are, their metabolism, and how they communicate with each other. So we talk about microbial ecosystems. Okay, so what are microbial ecosystems? They are systems that consist of all of the microorganisms that live in a certain space that is usually referred as a niche, a certain specific environmental location, and that work together, so not as single entities just opposed one to the other, or they just found themselves to stay in the same place but with no communication. No. The microorganisms in an ecosystem, they live in the context of each other, and they function and adapt their metabolism in the context of each other, and in the context of the biotic or abiotic factors that are found in that environment. So biotic factors refer, for example, to their hosts, if they are found within the gut microbiota, or if they are found in the environment, or in ocean dwellings, up in the clouds, other factors that pertain to the environment that can be alive or not, so biotic or not biotic, and that interact with this ecosystem in this niche. So obviously there's various degrees of complexity of this ecosystem, because they can be like very simple, be composed maybe of just two species. If they live together, it is an ecosystem. In the vagina, for example, the ecosystem is relatively simple compared to other locations in the human body. There is a reduced diversity and only a limited number of species, and that's fine for that ecosystem over there. There may be more complex ecosystems that are composed of hundreds of different species, or even 1,000 different microorganisms that reside there. So the way the number of these species, the number of the structure of the ecosystem itself, how it develops, and how it is structured, how it is located, and how it relates with its environment, these are all studied by the discipline of microbial ecology. So in this course, we are focused on microbiotas, so ecosystems that are associated with the human host, and in particular the oral and the gut microbiota. So just a little bit of terminology that may help clarify clarifying some misunderstanding that's often comes across also when reading papers in the field and so on. So microbiota, metagenome, and microbiome are three terms that we will use a lot in this course, and that you will encounter often when you read papers and literature that relates to this course, and maybe you have encountered them already for other coursework. So what is the microbiota? When we talk about this ecosystem, the microbiota refers to all of the microorganisms that reside within that specific niche. Okay, so within a defined environment, like the totality of the microorganisms. So it can be bacteria, but it's not only bacteria, it can be also viruses, it can be protozoa. So all the microorganisms within that niche constitute to the microbiota. The metagenome, on the other hand, is the collection of their genes and genomes. So obviously, every member of the microbiota, so every bacterium, every protozoan, every archaeon will bring its own genome, which will differ from the genomes of all the other components of the ecosystem, right? So when we look at the sum of all of these genomes, so at the totality of these genomes added together, that refers to the metagenome of that ecosystem, okay? So it's all of the genome of its components. And then we have the microbiome. So the microbiome is a more encompassing term. So it's not the synonym of microbiota. It includes the microbiota, but it also includes all of the other factors that are part of this ecosystem, including host factors, host-derived factors like metabolites, microbiota-derived factors, and so on. So here is a representation of the concept. I know you don't have this slide, and I can post it separately because I added it later, but I posted required reading on Quercus, and it's a manuscript that you can access through PubMed if you connect through your U of T account, and this is a figure in that manuscript, okay? So this provides like an overview of what we just said. So the microbiome encompasses everything. It includes the microbiome, it includes the metagenome, it includes the microbial elements like the protein, lipids, peptides, nucleic acids, and it includes the environmental factors like the conditions and signaling molecules that may be produced by the microorganisms themselves. So with the development of microbial ecology and the appreciation that human beings live in association with microbial ecosystems, and actually different regions of our body provide a specialized niche to different microbiotas, then here came the appreciation that maybe we should not consider ourselves like an organism, but maybe we should look at ourselves like supraorganisms that incorporate both our genes and our genome, and also the one of our microbial counterparts. And in fact, metabolic features that we derive from these added genomes are increased, because as we will learn in this course, these microbial counterparts provide us with a series of metabolic functions that we did not have to develop on our own. So their activity, obviously when we are in a condition of homeostasis and of symbiotic interaction with these microorganisms, their activity is complementary to our own metabolic activity, and therefore it's necessary for us to thrive in health. So a little bit of the history of microbial ecology and how we came to appreciate the importance of these communities of microorganisms, but also the fact that they are indeed communities and not, as I said earlier, just different microorganisms that find themselves in the same place. So this discipline has evolved throughout the years, and again, this slide is from the same paper that I put a reference on Quercus for you. So, you know, the developments in the field were really driven by technologies becoming available and allowing us to actually study these microbial counterparts and appreciate and learn about them. So in the very beginning of era of microbiology, which was born in the mid of the 1600s, right? It's considered that historically, I'm not sure there was already a PhD at the time. So I will say the scientist Anthony von Leeuwenhoek in Holland, he was the first to actually see microorganisms because he invented the first microscope. So he's usually referred to as the father of microbiology because with this instrument, he was able to actually see microorganisms. And the first that he saw were the ones in saliva, actually. So since then, obviously our knowledge has evolved and the evolution has been allowed by the evolution in the technology. So the technology progressed until more recently in the 2000s, right? We were able to use gene sequencing techniques and technology, and we were finally able to sequence all of these genomes of these microorganisms in this metagenome, even those that we had never cultured and even those that we didn't know they existed. Because now we didn't need to culture them to study their DNA. We just could extract the DNA from a complex sample, and even if the DNA is unknown to us, we are still able to sequence it, right? So this DNA sequencing really, when it became applied to the study of the microbiomes, it really the field made a big jump ahead. So in parallel with the development of the technologies and of our knowledge, we also came to appreciate more and more that these microbial communities have like a social character to them. So in the very beginning of the microbiology field, the first thing we learned about microorganisms were all about pathogens, right? So we were, the first thing we were exposed to, our first knowledge came from pathogenic agents of disease. So there was a disease and then a pathogen was identified, and there was this causal relationship with one pathogen and one disease, and therefore this idea that bacteria are bad to us because they cause disease. Right? As our knowledge developed, we also came to appreciate that in fact, yes, there are pathogens, but even the pathogens themselves could live in cooperation and together with other microorganisms. And the fact of living with other microorganisms may affect their own behavior, not only of the pathogens, but also that there were other microorganisms that are not pathogenic and that they're part of complex communities that are not necessarily causing a disease. And so we became to appreciate how these microorganisms within a single niche their preferred habitat or preferred environment. How do they communicate to each other? And we still have a lot to know about this. We know that they can secrete some chemicals and some metabolites that are helpful for them. They can use metabolites that they find in their environment that sometimes are produced by the host if they live in an association with the host. And we know that in response to these metabolites, there can be positive or negative responses and their gene expression can change. So we know that even within microbial communities, there is systems for microbe to microbe communication, right? Our cells communicate to each other, but also the microbial cells within a certain niche, they do communicate to each other. So there is this social aspect and this social idea about how these microorganisms interact within a niche. So we spoke briefly about potential ways and mechanisms that this microorganism can use to interact, and I said sometimes it has a positive effect, sometimes it has a negative effect, and sometimes it has a neutral effect, meaning no effect on none of the partner in the relationship. So usually that's exactly what we see. We can see an example of positive interaction, neutral, and negative. So for positive interaction, the three possible ones are mutualism, synergism, and commensalism. Okay, and for neutral interaction there is no effect on one another, and for negative interaction there is those four listed there. So for this course, I think the most important that we need to know exactly the meaning of these terms. So what is commensalism? So it's an association between two or more microorganisms and even species, across species, like it could be also human-bacterium interaction, that this relationship is not detrimental to any of the two partners and is usually beneficial to one of the two. So if we stick to our gut microbiota, many of the members of the gut microbiota are commensals to us. So they have with us that type of interaction. Or there can be a mutualistic interaction, so mutualism, that refers to an association that is beneficial for both partners. So again, in our gut, there are some bacteria, say bifidobacteria, for example. Some of them, not all, are in a mutualistic relationship with us because they produce metabolites that are useful for us, and we also, in exchange, provide them with an energy source for thriving, for example. And then there is amensalism, which, on the other hand, can have more of a negative connotation because it's the impact on one species on the other is negative, but there is no detectable impact of the second species on the first. Okay, this is amensalism. And parasitism is an association that is beneficial to one partner, but at the expenses of the host. So we have parasites, for example, in our gut that are at benefit from the interaction with us, but we do not, but it's at our expense, actually. And finally, there is pathogenicity. So pathogenicity refers to that first type of interaction that was described historically, right, in the 1800s. So the pathogenicity refers to the ability of certain microorganisms to actually harm their host. So there can be opportunistic pathogens that infect or cause disease only in a compromised host. So there might be microorganisms that are not pathogens for a person who is healthy, but on the other hand, in an immunodepressed person, for example, they may become pathogenic. And obligate pathogens that are pathogens that associate with the host only in case of disease. So for example, if we eat contaminated food with, I don't know, Campylobacter jejuni, then that will be pathogenic for everyone, but that bacterium, we ingest it with, in this case, a contaminated food. Can you hear me at the end of the night? Okay. Make some signs, my voice is always okay. Okay, so, yes.
Sorry, I didn't really understand the last slide. So opportunistic pathogens and obligate pathogens, they both cause infection in hosts that are sick. They're both unhealthy.
So they are both they are pathogenic, but the obligate pathogens are pathogenic for everyone. So, and these microorganisms, usually we don't carry pathogens, or I should not say that because maybe we have very low levels, but they are kept under control. But let's assume, like, we don't ingest a contaminated food, so we don't have that pathogen, right? But the moment these exogenous microorganisms is ingested and it is a pathogenic microorganism, then it will cause disease in any host. Like E. coli? E. coli O157H7, yes, not all the E. coli, but yes, for example, like the burger disease, hamburger disease.
So that's the obligate pathogen?
This is an obligate pathogen.
It is pathogenic for everyone?
Yes. The opportunistic pathogens, they become pathogenic only in certain conditions. So, for example, if the host, us, is unable to handle them. Like in our intestinal system, for example, we will learn we are protected by the intestinal barrier function, which has an immune component to it, which Dr. Fitton will tell you everything about that. So in some hosts, for example, immunocompromised hosts or immune deficient, then this barrier is not perfectly functioning. And therefore, in that case, an opportunistic pathogen who would do no harm to a healthy person with an immune system that is functioning, then it would become pathogenic for that person. Okay, so the discipline of microbial ecology also studies how these bacteria, these microorganisms interact with each other. And one aspect of this research is for us to visualize how this interaction occurs, because that helps us understand them better. So usually we construct these kind of networks that, like webs, that you will see in many publications and may come up in some of the slides later in this course, that illustrate, for example, in this case on the left, cooperation. So those nodes here would represent different types of microorganisms, different kinds of microorganisms, and these edges would represent their interaction. So usually it's not like a written rule, but it's more of a consensus in the literature, but you can find different colors. Usually if it's a positive cooperation, a positive interaction is shown with a green line. If it's negative, it's red. But again, like this, there is no rule for this, but that's most of the case. You will find these two colors. So this is how it's one way we have to represent this interaction. And then in the field, once we have identified which microorganisms interact positive or negatively, which other ones within a certain niche, then we may want to go into a more specific approach, like is visualized here, and understand in certain specific hosts how those interactions can be handled and what would be their physiological consequences. So I said earlier that we are supraorganisms and that microorganisms are associated with our body, and they are found almost everywhere. So there are many, almost everywhere in and out our body, you can find microorganisms, including your cavity intestinal tract, but also the urogenital tract, the skin, and to give like the major example. So obviously our body has had to develop the capacity to live, to coexist with these microorganisms, right? Without having any inflammatory reaction or any disease or infection that occurs, because we are talking about commensals. But at the same time, our body has also developed the ability to block those pathogenic ones that the body does not want to establish in or within the body. Okay. So this is a balance, like a homeostatic condition that has many factors that come into play, and that we will see throughout the various lectures in this course. So as I said in the beginning, it was technological advances that allowed us to understand and appreciate the complexity of this microbial ecosystem that are localized within the human body, right? So here is a slide that shows you where we were in 2008, which is about like the time when all this hype, right, because it became a hype, this microbiome research, was like starting, right? So as you can see, like these were the first human microbiome projects around the world. So they say the proliferation, these are papers in Nature. If you're interested, you can access it through the U of T library. So as you can see, like most of the funds were invested in the United States, and in Europe, especially in France, China, Canada also had a little bit, and Australia and Singapore. So now these projects have flourished and bloomed, and luckily they have expanded to more and more regions around the world. But still, at the beginning, these were the countries that started investing. So if you look at the literature, most of the manuscripts and most of the papers that are published are still coming from these countries. This is where most of the data are available, right? North America, Europe, China, Australia. And the interesting thing is that as research progressed and we were able to expand our studies across different geographies like Africa, South America, different countries all over the world, this really also made our knowledge grow, because we started to appreciate how many interactions that between host and microorganism that we had given for granted, discovered, and known based on this first microbiome project, in fact, depend on geography and on where people live and what they eat and what their environment is. So we always have to keep this in mind. The field is still so new, right? So as I was saying the first day, there are very few final answers here. And so the research is so progressing, and especially from the geography perspective, as more populations are included in these studies, we learn more and more on how things can work differently depending on ethnicity, depending on age, depending on where people live and what's the environment they're exposed to. Okay, so the Human Microbiome Project was the one that had as its aim to sequence the entire microbiome associated with humans, with human beings, and built on the concept that we already said earlier, that the gut microbial genome is a complement to the human genome, and that it provides all these functional potentials that we don't necessarily need if our microbiome is healthy and it contains all the functions that we need. So we know that there is about 1.3 microbial cells per every human cell. So not 10 to 1, as people used to think until a few years ago. But there are about 100 times more genes in the microbial metagenome compared to the human genome. So when we look at the human microbiome, yeah.
So what was the ratio that you were?
It's 1.3 to 1.
Thank you.
Okay, so if we consider the microbiota living in association with the human body, so all the different microbial, if we kind of put them together and we want to know which are the dominant phyla, these are the 4. Okay, so we are dominated. So these are 4 names that you want to remember, right? They will come up many times. You will hear them a lot, but you want to remember these 4 names. So actinobacteria and firmicutes, which are gram-positives, and proteobacteria and bacteroidetes, which are gram-negatives. These are the 4 main phyla that are found. They're not the only ones, okay? There's more than 20 phyla that we know of in microbiology, okay? But 4 are the predominant in the human body, not the only 4, but the main ones. So this was like a pioneer study. Yeah.
Sorry, this is the entire human body, right?
Not just the gut. Turns out that in the gut it's the same, but the relative abundances are different. Yeah. So you don't necessarily have to remember these percentages, okay? So so this was a pioneer study, was published in Science 2009. So they looked at different areas in the human body, in males and females, across different times, and they found that these were the 4 main phyla, and that there was speciation based on habitat rather than sex or time. So if you look at these pictures, that you will see them a lot in this course, every dot represents a sample. Okay? So a sample of that microbial community in a certain individual for that organ. So in this case, if we look, for example, at the green, each dot was a sample that was taken from the oral cavity, and each dot corresponds to a different individual. The light blue dots were taken from the gut. Again, every dot is from a different person who participated in the study. So each dot represents the data from the entire community, right? So in this time, they were doing 16S sequencing, so they were sequencing the 16S RNA genes. So that dot represents the sequences of all of the microorganisms found at that site in a specific person. And then with data analysis, and in this case it was principal component analysis in order to see the degree, let's say, the degree of relatedness between communities across sites and across individuals. Right? And we see those that cluster and group, those that group together are more similar to each other than to the other ones. And what they saw is that they grouped based on the location. So all of the sample from the oral cavity from all individuals grouped together. They didn't group by individual, right? We didn't find all the microbiome from the gut, the oral, the skin, and what I see, they look at the nostrils and the hair from the same person together and then from another person. No, they grouped based on the body site. Which kind of, now that we know it, kind of makes sense, right? Because each body site is a totally different environment, like in the gut, no oxygen, for example. On the skin, exposed to the external environment, right? They're completely different habitats. Yes?
I just want to ask, like, these communities, are they like also similar for people across the globe, like everyone, or just like North America or something, or no, are these like?
So this idea of the clustering by body site is supposed to apply everywhere in the world. Then maybe the microbiome of the oral cavity on someone living in South Asia is different from someone living in the Arctic, right? Maybe that you will see a separation, but the body size speciation is maintained. So that just reiterates this concept. So it was a follow-up study. They sequenced the different body sites, and each color corresponds to a different phylum. And obviously, you don't have to remember the composition. So this is just to remember the concept that the composition is different. Even though the phyla represented are the same, the relative abundances are different across the different organs. Okay, so in this course, we are focused on the intestinal tract, which starts with the mouth and ends with the anus. Okay? So, and the two largest and most diverse communities that reside within the human body are located, one in the mouth and one along the intestinal tract. Okay, so the first one that we are going to talk about is the oral microbiota. So this community, oral microbiota, the terms, refers to the entirety, to all of the microorganisms that inhabit the oral cavity. So they can be found at various sites. They can be found on the tooth surface. They can be found in the crevices between the tooth. They can be found on the mouth, on the tongue, the mucosa, and if people have implants or orthodontic work, they can be found also on those structures. These are the predominant organisms in healthy mouths, because I know that we tend to associate the presence of microorganisms in the mouth with something negative, but this is not necessarily the case. Some microorganisms that constitute the commensal oral microbiota are actually beneficial to us. Is when this community becomes either inflammatory or becomes cardiogenic, as we will see in a few slides, that then it becomes a problem and may cause caries, for example, or periodontitis and these diseases. Okay? But most of the bacteria exist in a symbiotic relationship with the host. So you don't have to remember all those names, but you may want to remember the Streptococcus and the Porphyromonas, because these are the pathogenic agents of some oral diseases. So, and as I said earlier, Anthony von Leeuwenhoek, and I'm sure that I'm not pronouncing his name correctly, is Dutch scientist that lived in between 1632 1723, and he invented a microscope, which this was the first microscope that he invented. And he looked at saliva, and he was the first to see microorganisms, and he used saliva as a sample to see them. Okay, so I said some, yeah.
Yeah, I know that like for like strep, there is like a period of like dormant when it's like stays in your system. That is it an opportunistic pathogen where it's waiting?
Which
for-
one? The one
like strep or like mono.
strep.
There's incubation period. So is that opportunistic because they're waiting for it, or like waiting for the?
No,
host to be
because
sick?
caries, the Streptococcus mutans will be a pathogen to everyone. It's when it's above a certain number, meaning that it produces enough acid to solubilize the mineral of your tooth. So
So it's multiply. So the incubation period is just multiplying in your system until.
yes, and actually it can multiply rather fast if the person, for example, has a high intake of sugars and no oral hygiene.
Thank you.
So these microorganisms live on teeth, and they form this biofilm, which we know as plaque, right? So that is a scanning electron micrograph of dental plaque that shows how there are many different microorganisms that are interspersed in those polysaccharides matrix, and altogether this constitutes this biofilm, which is a community of microorganisms that is called dental plaque. So it's a dynamic and highly complex because it's dynamic because its composition changes with time. It's not like in a fixed. Obviously, these microorganisms live and die, but also depending on what we eat or what our cells in the mucosa produce, which metabolize them, some will be favored over others. So it's a dynamic equilibrium, and it's a dynamic structure, and it's also highly complex ecosystem because there's hundreds of different species, and they live, and they also need compounds and products that are produced by the host in order to establish and to form this ecosystem, as we will see in a few slides. So, for example, when we refer to the plaque of the tooth, we call supragingival plaque when it's above the gum line, and subgingival when it is below. So I already said, so the dental plaque is a biofilm. We will see what is a biofilm momentarily. It includes about 800 bacterial species that grow on the tooth surface and are interspersed in a matrix of extracellular polysaccharides and salivary products, because as the plaque matures, some of these microorganisms are able to produce polysaccharides that are exopolysaccharides, so they are outside of the cells. And there are also salivary products that become entrapped, or there are salivary products on the surface of the tooth that facilitate the adhesion and of the bacteria to the tooth surface, and also the progression of the biofilm into the dental plaque. So dental plaque is a biofilm, and the way it follows the typical steps of formation of biofilms on solid surface, right? Because tooth is a solid surface, and so usually when when biofilm forms on solid surfaces, these are the steps that are followed. So now we are talking about the plaque, but for example, so last week you had that lecture on food microbiology, right? So you heard about milk being pasteurized, for example, or undergoing this high temperature treatment to control the milk microbiota, yes. So for example, in those pilot plants where milk flows in those metal pipes and then it's heated, sometimes there are biofilms that form on the surface of those pipes. So the way they form is exactly the same. So the main steps, not exactly the same obviously because the surface is different, microorganisms are different, but the steps of formation of a biofilm on a solid surface are the same. main steps. In the beginning, there is a very quick one, which lasts for a few seconds and which is still reversible. So there is this interaction which is not receptor mediated. It's a reversible interaction, absorption of the bacteria with the solid surface, but it's very short, right? Soon after, the attachment becomes irreversible, and these microorganisms are now on the surface. So at the beginning, there are a few, but as they grow and divide within hours or days, months, it depends on which microorganism, they all have different generation time, also depends on where they are found. The generation time, they will multiply, and this biofilm will grow until it becomes more complex, and some of these microorganisms are able to produce exopolymers, like in the case of plaque, exopolysaccharides, that contribute to create a matrix where more microorganisms can become entrapped, including those that would have not been able to attach in the beginning to the to the surface, right? Now that is more complex, and there are exopolymers that are often, in most cases, polysaccharides that offer new receptor sites to new microorganisms that now are able to attach and adhere to either the polysaccharides or to the bacteria that were already there, creating a more complex biofilm community. So the formation of plaque on tooth follows the same steps, except that step one, so this step one here is preceded by the formation of what is called the acquired enamel pellicle. Because on the tooth, the bacteria do not attach directly to the mineral which is called digrossiapatite, of which the tooth is made. Because the tooth, immediately after eruption, but also after professional cleaning, becomes covered with a layer of proteins that are derived from the host. And this layer is called acquired enamel pellicle. So the microorganisms attach to this pellicle and not directly to the tooth mineral. Okay? So what is the acquired enamel pellicle, or AEP for short? It's a biofilm, but it's an acellular biofilm. So there are no cells here. It's constituted of proteins, which is formed by proteins, also carbohydrates and lipids, yes, that absorb onto the enamel surface. Okay, so even before the bacteria come, this happens. So where do they come from, these proteins, carbohydrates and lipids? So they come essentially from four sources. They come from the salivary glands, from the oral mucosa, the microorganisms that are found in the planktonic phase. Planktonic means in solution, so that are in the saliva basically. And the gingival clavicular fluid, which is a fluid that is secreted at the crevice between the gingiva, and the tooth. Okay? So these are the four main sources of proteins and peptides that are released into the oral cavity and get absorbed on the tooth surface to form the AEP, the acquired enamel pellicle. So how does this happen? So it's a stepwise process, right? The formation of this pellicle. So you have subsequent layers of protein and peptides that adhere to this tooth to form these pellicles that then, ultimately, when it's mature, it's hundreds nanometers thick. So these, some of these proteins that are produced, like as we saw, right, the gingival clavicular fluid, or derived from the salivary glands or oral mucosa, or the microorganisms, those are examples of some of these proteins. The histatines, the proline-rich proteins, staphylococcus, alpha amylase, and so on. Some of them, in their intact form, then they're shown in purple here, they absorb into the enamel in their intact form. Others are cleaved before absorbing. They are cleaved by enzymes in the saliva, and they absorb as cleaved proteins. And as such, they can either absorb on the enamel itself or they can absorb onto other intact proteins that were already there. So it's a stepwise process that leads to the formation of this pellicle. So once we have this pellicle, then the other steps can occur for the formation of the plaque biofilm. Okay? Then the microorganism come, not any microorganism is able to initiate the formation of plaque. Those that can are called the first colonizer. So this first colonizer, there is first a physicochemical interaction with the surface, with the tooth covered by the AEP, right? Like the phase 1 and 2 that we saw in the biofilm formation with electrostatic forces that play a role here, before a receptor-mediated attachment, which is a more specific recognition with a ligand that needs to be present so that only those specific microorganisms can attach through this, to this mechanism, right? Once the first or primary colonizers are established on the tooth surface, on the AEP covered tooth surface, then the so-called second colonizer comes, which can attach, sometimes is just a position, but in many cases it's co-aggregation, so they recognize receptors that are expressed by the first colonizer. So they need those receptors to be there. Therefore, they couldn't establish first if the primary colonizer weren't there already. And then in step 4, similar for any biofilm, there is multiplication of these cells and then the production of exopolysaccharides. That is very important step in the case of plaque, because it's that complex matrix that allows the different microorganisms to be entrapped and the plaque to grow. Okay, so this is just a different representation with some examples. For example, streptococci are among the primary colonizer. So this is the tooth surface, this is the acquired enamel pellicle, and microorganisms like streptococci that are among the first colonizer. And then as the plaque develops and the biofilm grows, different microorganisms come in, like actinomyces, and then the second colonizer, like those that you see there. And the plaque grows. So because this is an ecosystem, it is characterized by certain metabolic relationships, right? So is what we have already said. So for example, within the plaque, there are lactic acid bacteria that produce lactate, and then there are microorganisms like the Veillonella, for example, that utilize lactate and produce, for example, menaquinone and analogs that other microorganisms like Prevotella or Bifidobacterium need to thrive. So there's a cooperation between the different members of the community. So it's really a community of microorganisms that have these symbiotic interactions. So factors that can affect the growth of the biofilm, I think you can think of what this could be. Obviously, nutrients availability applies to anything, to any biofilm, pH, oxygen, because depending the microorganisms may be facultative anaerobes, they may tolerate oxygen, they may be aerobic or anaerobic. So depending on how much oxygen is available in different locations of the biofilm, it may favor different microorganisms. Aggregation factors, host factors, obviously, yeah?
We need to memorize all the relationships in this.
No.
Okay.
No, you don't need to remember. But the lactate, you may want to remember because this comes up a lot. The Veillonella as a user of lactate can be a good example of a relationship that you can remember. Like Streptococcus would produce lactate and Veillonella would use it. Yeah. This is a common one. No, you don't have to remember. And all of these factors therefore intervene, and obviously plaque across different individuals is not the same, right? Because there are many, many factors that determine its composition and growth. Also, biofilms can form at different sites within the oral cavity. We saw earlier, so depending on where it is, the composition may be different, the steps would be similar, except that the acquired enamel pellicle is the one that forms on the tooth, right? Because the tooth is the mineral, so it's a different substrate from the soft tissues that are found in the rest of the mouth. So, and as I was saying, not to forget that when it's under control, the oral microbiota does have some positive effects. So it provides, for example, a barrier, like a physical barrier, blocks sites of attachment for pathogens, for example, because they are already occupied, and so a pathogen may not be able to infect. They contribute to host nutrition through the synthesis of vitamins, still to be confirmed, because yes, they produce vitamins, also bacteria in the intestinal tract, though for those in the mouth specifically, the availability of the vitamins and the effect on our vitamin status requires more research. They contribute to immunity by inducing low levels of circulating and secretory antibodies that may cross-react with pathogens, yes. And they may produce substances like bacteriocins or short-chain fatty acid that counteract pathogens again. So there are some beneficial effects. Though this community, in response to host factors again, or to nutritional factors, anyway, environmental conditions, may change its composition and its functionality and may become dysbiotic. So it is not a health compatible or eubiotic composition anymore, it's dysbiotic. And in this case, it is associated with disease, and different dysbiotic oral microbiota are associated with different diseases. So the two main ones that we want to remember are those in red here, the Porphyromonas gingivalis and the Streptococcus mutans, which usually are not found in healthy mouths. So this is what these two bacteria look like. So the Porphyromonas gingivalis is a gram-negative anaerobic, and it is the main cause of periodontitis. So it is an inflammatory disease that that is the consequence of the invasion of the epithelial cells by this pathogen. The Streptococcus mutans is a gram-positive and is the cause of caries. So dental plaque is the etiological agent associated with dental caries and periodontitis. If then these two bacteria become in numbers that are high enough to cause the disease. So for example, if the plaque becomes cardiogenic, meaning if it becomes populated with Streptococcus mutans, which is not supposed to be there in a healthy mouth. So how does this happen? So because, for example, there is overgrowth of microorganisms that produce acid, but also that are able to thrive in acidic conditions. Usually when the pH lowers, microorganisms are not able to cope with the lowering of the pH. But now these pathogenic streptococci may be more able to withstand lower pH, and they become in higher numbers, and this usually happens following high intake of sugars, especially often, like at a high frequency. So this results in dental caries, which is the most common tooth disease. So hydroxyapatite, this compound here, is the mineral that constitutes bone. So when the pH lowers below 6, as a consequence of the production of lactic acid by the bacteria in the plaque following frequent exposure to sugars intake, okay, frequent exposure to sugar intake, no or low oral hygiene results in these microorganisms proliferating and producing acid. So because now the acid becomes like a lot, it may lower the pH. So if it lowers the pH below 6, then the hydroxyapatite solubilizes and goes into solution. So the tooth starts dissolving, which is what caries consist of. On the other hand, at the higher pH, the remineralization is favored. So the equilibrium of this reaction is towards the left. So there are many bacteria, different ones that can be found in cryogenic plaque, and these are some names that you don't have to remember, just to give you an idea. It's not just Streptococcus mutans. There are many species that can resist acidity, so survive at low pH, like all the one listed here, including, I just want to point out, lactobacilli and bifidobacteria, right? That we tend to associate, and they certainly are associated with beneficial bacteria, right? Especially in the gut, they are thought to be beneficial. But let's not forget that they may also be found sometimes in the mouth in a situation of cryogenic plaque. Okay, on the other hand, periodontal disease, it's an inflammatory disease, and it's an inflammation of the gingiva. So this can have two steps. In gingivitis, we have an inflamed gum, right? In periodontitis, the disease has progressed, and there is a plaque-induced inflammatory response that ultimately may lead to tooth loss. And this is the one that is driven by that other bacterium, the Porphyromonas gingivalis. So beyond these two main oral diseases, there are other detrimental effects that the oral microbiota may have, especially if the composition is switched towards certain species that are not compatible with that. So but oral bacteria have been implicated in all of these conditions. Endocarditis, bacterial endocarditis, abscesses of the alveolar bone, lung, brain, or the extremities. This can happen, for example, if there are injured gums or gingiva, then bacteria can gain access to the circulation and travel to other organs. They have been associated with preterm low birth weight and also with heart disease. So a few years ago, a paper appeared that microorganisms in the oral cavity may also be associated with the metabolic syndrome. As this was, I think this was the first study where they found that certain bacteria were present in different abundances in people that with or without obesity. And so highlighting potentially a new line of research where the oral microbiota may also play a role in the obesity disease, or maybe it's a consequence of this disease. But a follow-up study was performed where this time they were able to look at the entire salivary microbiome, not just a few specific species, and in fact comparing samples derived from normal weight participants and participants with obesity, they were able to see a separation between the microbiome in the two groups, showing in fact a distinct character between these two microbiotas, not only at the compositional level but also at the functional level. So with this, maybe we take a 5 minutes break before we go into the next part. So wait until 2:15.